All right, in this next video, we're going to talk a little bit about the next step in going from a DNA molecule to an expressed protein or a gene being expressed to make a protein. And this process is called translation and involves the ribosomes where protein synthesis occurs. So it's all based on the genetic code. So the genetic code is read in a sequence of 3 bases called codons. And the way they figured this out was pretty simple. They knew there were 4 letters, A, C, T, and G, or in RNA it's A, U, C, and G. And those 4 letters had to code for the 20 different kinds of amino acids. So they started looking, well, if each letter is an amino acid, we can only make 4. If we put it in groups of 2, we could make 16 combinations. That's still not enough. If we put in groups of 3, there's 64 possible codon. That's more than enough to code for all 20 different amino acids. So there's some repetition in the genetic code. So a codon is a group of 3 nucleotide bases in messenger RNA that specifies a particular amino acid to be put into a particular protein. So here's the AUG is the first codon of all proteins. It's what we call a start codon. There's the next one, and then there's the third one. And if this was a real gene, there would be lots of these together. So this is the genetic code. There's several different ways to look at this, and this is one way to show all the possible amino acids produced by all 64 possible codons. And so the way you read it, you start in the center with the first letter, then you move to the second ring for the next letter, and then the third ring is the outer letter. And you can see, then read on the outside ring the actual amino acid that would be produced from that particular codon. So for instance, here we've got C. A, C codes for histidine. There's also, I mentioned AUG is the start codon for all protein synthesis. There's also 3 stop codons. You can see them here in the table. So this process of going from taking a mature messenger RNA and using it to build a protein is called translation. So we're going from a nucleic acid message to a protein message. So we had replication, which is copying the DNA, transcription, which is DNA to RNA, and then translation, which is RNA to protein. So it involves all 3 types of RNA. It involves the messenger RNA, the transfer RNA, and the ribosomal RNA. So this all happens at the ribosome. So translation starts when a ribosome attaches to a messenger RNA molecule. Then the first tRNA molecule comes in, brings its amino acid. It binds codon to anticodon, so the base pairs are complementary between the tRNA and the messenger RNA. And then another one comes in, they form a bond called a covalent bond, a type of covalent bond called a peptide bond between the amino acids, and the ribosome simply begins moving down the messenger RNA until it comes to a stop codon when translation stops and everything breaks apart and you've got a produced protein. All right. So you can just see kind of the process here, how this works, and the ribosome is moving down the messenger RNA. As it moves down, the old tRNAs that have donated their amino acids leave. New tRNAs for the next codon come in, bonds are formed, and we just continue the process till the stop codon happens. So once it gets to the stop codon, the ribosome, the messenger RNA, and the tRNAs all break apart, and you've got a polypeptide, which becomes the protein that gets released. So there's the stop codon, there's the messenger RNA, there's our polypeptide. So like I said, all the 3 major forms of RNA, messenger RNA, transfer RNA, ribosomal RNA, are involved in the process of translation. So just to review the functions here of the 3 ribosomes, we talked about these in the last video. So here's kind of an overview of the whole process of making a protein. Starts with the DNA strand, transcribed into messenger RNA, goes out to the ribosome, and we use the messenger RNA, the transfer RNA, and the ribosomal RNA and the ribosomes to build the polypeptide. So there's something called the central dogma of molecular biology that describes the way that information flows from DNA to protein. So this is what it looks like. You've got replication, which is copying the DNA, transcription, which is DNA to RNA, translation, RNA to protein, and there's also something called reverse transcription, which is used by viruses and by scientists in molecular genetics to make an RNA copy, or use RNA to make a DNA copy of that, and we'll talk about why that's important in a little bit later video. So when a gene or a segment of DNA code is used to build a protein, we say that gene has been expressed. Now, not all genes get expressed in all cells, especially if we're talking about a multicellular thing because of specialization. You know, in a muscle cell, the genes for actin and myosin expressed a lot. Gene for hemoglobin, which is for carrying blood, oxygen blood, not so much. Whereas in a blood cell, you get lots of hemoglobin expression, not a lot of actin and myosin expression.